We started talking about the synthesis of non-essential amino acids last time. I told you that they can be grouped into families based on the precursor molecules that they use to synthesize these molecules, these amino acids. And we already discussed the synthesis of serine and glycine from 3-phosphoglycerate, so an intermediate of the glycolytic pathway. And we saw that for the conversion of serine to glycine, we need this interesting cofactor tetrahydrofolate. And I talked about the different forms that tetrahydrofolate can take and what one carbon units it can actually be bound to. So what we are going to discuss now briefly is how serine can be converted to cysteine. And here's a classic example where it really depends on what organism you're dealing with, because if we compare, for example, bacteria with mammals, it's actually quite different. If we look at what happens in mammals, we have a condensation reaction between serine and an amino acid called homocysteine. So again, this is an example of an amino acid that is not found in proteins, so it's not one of the 20 standard amino acids, but it's obviously a naturally occurring amino acid. It's called homocysteine because it's structurally similar to cysteine, the structure you see here, but has actually one methylene group more in the side chain. That's why it's called homocysteine. Okay, and this homocysteine is produced from the degradation of methionine, as we are going to discuss in a moment. Okay, so homocysteine is actually quite an interesting molecule. This condensation reaction here that leads to the production of this intermediate here, cysteine, is catalyzed by a synthase. It also has pyridoxal phosphate in the active site. Again, here pyridoxal phosphate serves to bind substrates, and we have a condensation reaction between the sulfhydryl group in this homocysteine and the hydroxyl group in serine to actually form a thioether bond between these two amino acids. And then this cysteine is cleaved in such a way that the sulfur, which previously was in homocysteine, ends up in this moiety that came from serine to form cysteine and alpha-ketobutyrate. After we also remove this alpha amino group here with the cysteine gamma lyase. Okay, so first this moiety has the sulfur, we form a thioether bond, then we cleave here, and the sulfur actually ends up in cysteine. So in bacteria it's different. I don't have any slides for them, but there we have actually an incorporation of a sulfite into the side chain here of serine. So I think it's serine. So it's quite different actually. Okay, so let's talk a little bit about homocysteine. And homocysteine is intimately related to another cofactor that can be utilized to transfer one carbon unit. So again, we talked about biotin as a cofactor that is capable of transferring carboxyl groups. Last time we talked about tetrahydrofolate that can transfer one carbon unit in a variety of different forms. We saw this whole pool of tetrahydrofolate derivatives last time, and now we are going to talk about a third cofactor that can be utilized for transferring one carbon unit, and it's S-adenosyl methionine. Okay, so this is also an important cofactor, and we see that it is composed, as the name implies here, of a methionine. Okay, This is methionine, the standard amino acid, which is linked via covalent bond directly to the 5' carbon of the ribose, which is linked to an adenine. So this is just the nucleoside adenosine. If we had a phosphate here instead of the methionine, this would just be AMP, okay? But we are lacking any phosphates here in this structure, and therefore we refer to it as a nucleoside. And since we have adenine here as our nitrogenous base, it's just an adenosine. Okay? And we are going to talk about the synthesis and degradation of nucleotides a little later, and I will also give you a bit more background information about the nomenclature of nucleotides. So here is a classical adenosine nucleoside linked directly to methionine, and therefore it's called S because it's linked to the sulfur, adenosine methionine. Okay, so this cofactor can actually be used by a number of enzymes to transfer a methyl group, this methyl group. Okay, and we will also see probably later today that, interesting enough, this cofactor is also utilized in the synthesis of so-called biological amines, polyamines in particular. And in this case, what is being transferred from this cofactor is a propylamine residue. Okay, so this excuse me, it's difficult. Okay, S-adenosyl methionine is not only used, strictly speaking, for transferring one carbon but also for this propylamine residue, as we'll see. Okay, but usually we just hear about this metal. So how is it being produced? Okay, it's produced from ATP and methionine, which is not astonishing, but interesting as far as all three phosphates in ATP are actually removed here, okay, to form a covalent bond between this 5' carbon and the sulfur. We cleave off phosphate and pyrophosphate, which will also be cleaved to two inorganic phosphates. And we form, with the help of methionine adenosyl transferase, this cofactor S-adenosyl methionine. And then this can be utilized by a variety, it says, of methyl transferases to incorporate methyl groups in appropriate target molecules. And I just give two prominent examples here. Synthesis of phosphatidylcholine, okay, which we, for example, primarily need for the synthesis of membrane lipids, okay, results from the methylation of phosphatidylethanolamine. Okay, so in choline we only have 3 methyl groups linked to the amine of ethanolamine, and they all come from this S-adenosyl methionine. And then, as we are going to see, epinephrine or adrenaline is actually synthesized from norepinephrine by utilizing a methyl group from S-adenosyl methionine, adding it to epinephrine. That's something we're going to see probably later today. So what happens if this methyl group here, shown in red, conveniently enough, is transferred to appropriate acceptor molecule by these transferases, is that we are left with an adenosine that is linked to homocysteine. Okay, so just look at the structure again. This was homocysteine. Okay, now we have the sulfur linked to adenosine, like here. Remove this methyl group, and then a hydrolase can cleave this bond between the homocysteine and our adenosine and release homocysteine. Okay. So we can actually utilize this homocysteine to produce methionine, and so we have to methylate it again, and this is done by methionine synthase, which requires cofactors that are capable of transferring methyl groups. And here we can either use tetrahydrofolate or actually methylcobalamin. We talked about deoxyadenosyl cobalamin when we talked about the degradation of odd-numbered fatty acids. You may remember that. There's actually a variety of that where there's a methyl group, and this methyl group can actually transfer here from cobalamin, for example, to methionine, homocysteine to form methionine. So what you have to understand here is that in order to convert homocysteine 2-methionine, we need to have sufficient concentrations of tetrahydrofolate or cobalamin. Okay, so if we have insufficient amounts of this in our body, we may not be able to sufficiently synthesize methionine from homocysteine, which would lead to increased concentrations of homocysteine in our body. Okay? And it has been reported some time ago that elevated homocysteine concentrations might be linked to cardiovascular problems and strokes and things like this. For some time, it was recommended that you take up tetrahydrofolate on a regular basis to prevent elevated homocysteine concentrations. Now, I actually looked into this a few years ago again and saw that the American Heart Association does recognize that elevated homocysteine concentrations are linked to things like heart attacks and strokes, but they, at least 4 or 5 years ago, they did not recommend to actually take up additional vitamins for this purpose. Okay. Reducing but be aware of that. If you hear about this, that's where it actually comes from. Okay, so I said that we would only discuss non-essential amino acids, or synthesis of non-essential amino acids in this class, and our discussion of the other non-essential amino acids will be rather short because we pretty much know already how they are being synthesized. So oxaloacetate, the intermediate of the citric acid cycle, can be easily converted into aspartate. We talked about this before. This just requires transamination reaction. Then aspartate, if it takes up an amino group in the side chain to produce an amino group, is converted into asparagine. All the other molecules that are produced ultimately from oxaloacetate are essential amino acids. We don't synthesize them. Similarly, the only non-essential amino acid that is produced from pyruvate is alanine, and we know that it can be produced in a transamination reaction, so we don't have to really talk about this anymore. What we're going to spend some more time on is how amino acids are being utilized to synthesize other important biomolecules, and we will actually talk first about the synthesis of heme, finally, and its degradation process. So I mentioned probably a dozen times before that heme is actually produced from succinyl-CoA and glycine. Okay, here in this representation shows acetate, but this acetate comes from succinyl-CoA. We see that all these atoms here, with the exception of the iron, of course, actually stem from these two precursors, the standard amino acid glycine and succinyl-CoA intermediate of the citric acid cycle. So we're talking about heme. So how is heme actually synthesized? It's synthesized either in erythroid cells, so immature red blood cells, or in the liver, and the synthesis takes place partially in mitochondria and partially in the cytosol. And there are some intermediates that can be transported across the inner mitochondrial membrane. So succinyl-CoA is produced in the citric acid cycle, and then we have an enzyme synthase that catalyzes the reaction of succinyl-CoA to glycine. Okay, so glycine actually is decarboxylated. We see this here, and this alpha carbon of glycine attacks our thioester bond in succinyl-CoA and produces this acid here, delta aminolevulinic acid, which is pretty much a pentanoic acid that has an amino group and a ketone here. Now, what I would like you to pay attention to here is the similarity to the sphingolipid synthesis that we discussed last time, I believe, where we had this reaction of serine with palmitoyl-CoA to synthesized this precursor of sphingosine. We also had a decarboxylation reaction there. So this decarboxylation produces a carbon ion, which is a good nucleophile. The carbon ion then attacks this thioester bond here in palmitoyl-CoA. And similarly here in this reaction, we have a decarboxylation reaction, and this carbon actually attacks here to produce delta aminolevulinic acid. So this compound is then transported out of the mitochondrial matrix into the cytosol, and two of these molecules are utilized by porphyrinogen synthase to produce a derivatized pyrrole ring. Okay. Remember, hemes are based on tetrapyrrole systems. So here we actually have two delta aminolevulinic acids being utilized to produce a pyrrole system that has propionic acid residue, acetic acid residue, and then this methylamine here. And four of these structures are then used by these two more enzymes here to produce a tetrapyrrole system, which as substituents has acetate residues, that's meant by the A here, propionic acid residues, and has still four methylene bridges here bridging these four pyrroles. Then we have decarboxylation reaction catalyzed by a decarboxylate. I won't go into these names, I'm not going to remember these anyway, I suppose, to actually convert the acetate residues here, A, to methyl residues, which we have then in our final product also. Then this compound is transported back into the mitochondrial matrix after we remove two more 1 carbon units to convert propionic acid, or actually, where does this happen? I'm not quite sure where this happens actually. I think this, not sure what is transported, probably this protoporphyrin line, I'm not quite sure. But we remove two 1 carbon units to convert two of these propionic acid residues into vinyl residues, and then an oxidase actually converts these methylene bridges, bridging the four ring systems into methynyl groups. So we have these double bonds here also, and then ferrochelatase actually incorporates the iron, the central ion here in heme, to produce heme. Okay. So, trust me, I'm not going to ask any details on this in any exams, but just have a general idea where this happens, how it happens. Okay, so heme is then, let's say, if we have heme in red blood cells, it's utilized for transporting oxygen, hemoglobin, and then when it's time for these red blood cells to be removed from the system, the protein is broken down, and then also the heme is broken down. And how this happens is shown on this slide. This is actually primarily happening in the spleen first, or starts in the spleen, in macrophages in the spleen, where we have oxygenase called heme oxygenase, which cleaves the bond between this A pyrrole ring and the B pyrrole ring to produce a linear tetrapyrrole. Interestingly enough, this reaction not just releases the central ion, the iron that we have in there, but also produces carbon monoxide. Okay. So just think about this carbon monoxide. We have all these detectors in our bedrooms to warn us about carbon monoxide, and here we ourselves actually produce carbon monoxide in a biochemical reaction, metabolic reaction. But these concentrations are not significant. So what then happens is, what we produce in this process, a compound called biliverdin, and all these many of these degradation products actually have the name Billy in their name, okay, bile, and indicate also the color, kind of, okay. So this is biliverdin. verde meaning green, right? And this biliverdin is then reduced by reductase. So this double bond here between ring C and D is reduced, and we produce bilirubin, okay, which actually is not quite as red, I suppose, as the name implies, but is described more as orange-yellow or something like this. This is a quite hydrophobic compound. It's not soluble in blood, but is transported to the liver, actually bound to albumin. So we heard about albumin being able to transport a variety of hydrophobic compounds. We talked about fatty acids. Also the amino acid tryptophan, for example, is transported bound to albumin. Okay. So we have bilirubin now in the liver, and in the liver, let's say the attempt is being made to make this rather hydrophobic compound more hydrophilic. And this happens in a glucuronidation reaction where glucuronic acid, okay, so remember structures of uronic acids, we have a monosaccharide that has a carboxyl group with carbon number 6. Okay, here we're talking about glucuronic acid, so it has a carboxyl group with carbon number 6. And I may have told you that glucuronic acid is utilized in so-called phase 2 reactions to make rather hydrophobic compounds more hydrophilic, because we attach not only a sugar but even a negatively charged sugar, so it's very polar. And this is also done with bilirubin, so we attach two glucuronic acid residues in this glucuronidation reaction to produce this bilirubin diglucuronide. Okay. Now this molecule is more hydrophilic, is actually secreted into the intestine. Okay, and you would say, no, it's hydrophilic enough that it can be secreted, but it's still hydrophobic. But actually what happens is that microbes, or microorganisms I should say, in the large intestine like to cleave off these sugar residues and produce bilirubin again. And bilirubin then, to a large extent, is taken up again, reabsorbed into the system. Okay. But some of the bilirubin can also be actually converted by microbial enzymes then in this compound, urobilinogen, okay, which is also partially reabsorbed and transported to the kidneys, where it's converted into urobilin, okay, which is yellow and contributes to the coloration of urine also. Or this urobilinogen, after it's reabsorbed, oh no, not after it's reabsorbed, in the intestine, excuse me, can be converted also by microbial enzymes into stercobilin, which is brown. Okay? So very, very colorful chemistry here that happens, which can also occur in one way or the other, actually when you have a bruise and red blood cells are actually destroyed. Let's say you bump your arm or so, you have a bruise, and then you have red blood cells being lysed in a certain location where you actually bumped yourself, and then the heme can actually be degraded then at this location, and the changing colors of bruises actually are manifestation of events like this that occur. So let's now talk about another class of compounds that are derived from amino acids, and this is again a rather diverse group of molecules that have a multitude of different functions. What they have in common is that they all are amine-containing compounds that have biological functions, and for that reason are just called biological amines. And there is really no good way of further classifying these compounds. I found one way of classification in a German book actually at one point, and it classified biological amines based on the structures they contain. So there are, for example, imidazole-based biological amines, or indole-based biological amines, or catecholamines, which are derivatives of a dihydroxybenzene structure. Okay, there are a number of, I guess, familiar compounds among biological amines, like carnitine. We talked about the role of carnitine actually in fatty acid degradation. I just mentioned epinephrine, norepinephrine. You may recognize histamine, okay? Acetylcholine, for example. And then there are a couple of names that you may not have expected, like mescaline, for example, a psychedelic compound. These are all biological amines. So, what we are going to talk about first are some biological amines that are synthesized in a very straightforward manner, let's say. So here we have the most important inhibitory neurotransmitter, gamma-aminobutyric acid, or GABA. And you can just deduce the structure from the name. So it's based on a butyric acid, so 4-carbon dicarboxylic acid, which at one end has an amino group. And this compound is readily synthesized in a decarboxylation reaction from glutamate. Okay, and this happens with the help of a glutamate decarboxylase, which also has pyridoxal phosphate as a cofactor, never mind. Similarly, if we utilize an appropriate decarboxylase, histidine decarboxylase, we can decarboxylate readily histidine and produce histamine. Okay, so histamine, you may know, actually is a vasodilator and is released in allergic or inflammatory reactions, but it also stimulates the production of acid in the stomach. Okay, so another interesting biological amine is serotonin. This structure here, which has a multitude of functions. I just listed a few. So it's found in the intestinal wall, where it's involved in regulating the motility of the gastrointestinal tract. It also induces a constriction of blood vessels and also has a number of activities in the central nervous system. So it, for example, is involved in controlling sleep. It's important for memory and learning, but also controls appetite. And if you look at the two steps that actually produce serotonin, it involves decarboxylation. Okay, so this is a common theme in the production of these biological amines. We utilize an amino acid at one point. Sooner or later, we will have a decarboxylation reaction. Okay, so what we have first, however, in the production of serotonin is a hydroxylation of the indole ring of tryptophan to produce a 5-hydroxytryptophan. This is done by a hydroxylase, which utilizes not only molecular oxygen, but a cofactor known as tetrahydrobiopterin. And this cofactor you actually should have heard about, or you have heard about in Niati's talk. You mentioned that, right? Because it's a cofactor, oops, where are we? That is also utilized in the production of tyrosine from phenylalanine. Okay, so here's the structure of biopterin, tetrahydrobiopterin and dihydrobiopterin. And tetrahydrobiopterin is actually utilized also by phenylalanine hydroxylase to convert phenylalanine to tyrosine. I told you last time when we looked at the list of essential versus non-essential amino acids that tyrosine is considered a non-essential amino acid because it can readily be produced from phenylalanine hydroxylation reaction. So here we see it again. I think Niati actually showed this. But then again, some people argue that, okay, it requires the essential amino acid phenylalanine, so is it really non-essential? But it's a matter of definition. So if we lack this enzyme that carries out this hydroxylation in and the production of tyrosine, we learned from Niati that it may lead to phenylketonuria, okay, which may lead to brain damage. So the same cofactor is also utilized here, in the hydroxylation of tryptophan to produce 5-hydroxytryptophan, and it's also utilized in the production of catecholamines. I'll come back to that in a moment. I just wanted to show you the similarity between the structures of tetrahydrobiopterin and tetrahydrofolate, which we saw in more detail last time. So they're structurally quite similar. So let's take a look at the synthesis of catecholamines. Okay, so catecholamines all contain this bis-hydroxylated phenyl ring here. So we have to start, actually we do start with a hydroxylation of tyrosine. So tyrosine is a precursor molecule for all catecholamines. The hydroxylase that is utilized also utilizes tetrahydrobiopterin molecular oxygen to incorporate an additional hydroxyl group in tyrosine. So phenylalanine is first hydroxylated to tyrosine, and then tyrosine hydroxylase can add another hydroxyl group to produce this dihydroxyphenylalanine, okay, which is also called DOPA, and since it stems from an L-amino acid, or is an L-amino acid, it's often referred to as L-DOPA. And L-DOPA not only is the precursor here for these other catecholamines, dopamine, norepinephrine, epinephrine, but interesting enough, it's also the precursor for melanin, so your skin, eye pigment, okay, hair pigment. And insufficient quantities of dopamine, actually, which is a neurotransmitter that is produced in substantia nigra in mid-brain from L-DOPA, have been related to Parkinson disease. And for that reason, L-DOPA is actually used as a drug to treat Parkinson, because L-DOPA can be, can cross the blood-brain barrier, and then in the brain can be converted into dopamine. Okay, so if you have sufficient quantities of dopamine, it's fine, but if you don't, you may actually add L-DOPA. And this conversion of L-DOPA to dopamine then includes, or is based on a decarboxylation. So again, in order to produce these biological amines from amino acid, we have a decarboxylation, and it actually happens at this level where L-DOPA is converted to dopamine. Dopamine in other tissues can then, or in other cells, can then also be converted into norepinephrine, epinephrine. In order to go from dopamine to norepinephrine, if you compare these structures, we need to add a hydroxyl group here in the aliphatic moiety of dopamine. This is done with a hydroxylase that utilizes ascorbic acid, vitamin C as a cofactor. Okay. And norepinephrine is produced also in the brain, but also can be produced in the adrenal medulla and leads to arterial contraction, decreases peripheral circulation, and stimulates lipolysis in adipose tissue. Norepinephrine can then be converted to epinephrine by actually methylating norepinephrine here, okay, to produce epinephrine. This is done with one of those methyltransferases that utilize S-adenosylmethionine as a cofactor, as I indicated previously. So this is one of the most prominent examples where S-adenosylmethionine is being utilized. And epinephrine is produced in the adrenal medulla and has also a great variety of different functions, okay, contraction of smooth muscle, relaxation of others, increase of heart rate, what we all have heard about, but also stimulates breakdown of glycogen in the liver and muscle, and also stimulates breakdown of lipids and adipose tissue. So the functions with regard to, let's say, the breakdown of lipids in fat tissue and also glycogenolysis in liver are analogous to what we discussed for glucagon activity. Okay, so these hormones bind to appropriate receptor molecules, and then via a single transduction cascade, we have an increase in CAMP, and CAMP activates CAMP-dependent protein kinase or protein kinase A, and then phosphorylates targets. Okay, so this is analogous to what we discussed for glucagon. Okay, so now let's come to polyamines, and polyamines are compounds that the name implies that contain more than one amine, okay, have certain biological functions. And the polyamines that we are talking about here are primarily spermine and spermidine, okay, these two. You could argue that putrescine is also polyamine, I guess it is. And this putrescine here is actually produced from ornithine, so this was an intermediate of the urea cycle. Okay. Remember, the structure is structural analog to lysine, lacks one of the methylene groups in the side chain, otherwise it's identical. And if we decarboxylate this compound, we produce putrescine. So it's diaminobutane. Okay. This is done by an enzyme, ornithine decarboxylase, which is actually quite short-lived, as far as I know. There's a half-life of just a couple of minutes or so. And it leads to the production of putrescine, which can then be converted to spermidine and spermine. If you compare the structures of putrescine, spermidine, look at this blue box here, and spermine, look at these two blue boxes, what has been added to putrescine are propylamine residues. Okay, so 3 carbon units with an amine, and this propylamine actually stems from acetoadenosylmethionine. That's what I was referring to earlier. Acetoadenosylmethionine can not only be utilized for transferring methyl groups, but can also be utilized in these reactions that lead to the production of our polyamines, spermidine and spermine. What happens here is, okay, that acetoadenosylmethionine is first decarboxylated. So interesting again, okay, even for the transfer here of our propylamine, we decarboxylate our starting material. And then this propylamine residue is transferred by propylaminotransferases to our intermediates here. Now, polyamines have also a number of functions that are not completely understood, but what is known is that they are required for the packaging of DNA. We are going to start talking about genes and chromosomes and things like this probably next week, and I'll also talk about the packaging of DNA. What you have to understand is that chromosomes are single DNA molecules that are associated with proteins, also polyamines and cations, and these single DNA molecules, actually, if you fully extended them, can be quite long, not that long, but that long, actually. And we have chromosomes ourselves, okay, DNA molecules, single DNA molecules, that are about 2.5 inches long or so. And these long molecules actually have to fit into the nucleus of a tiny cell. So what has to happen is that this long molecule is actually packaged neatly and tightly enough they can actually fit into a cell. And for this packaging, these polyamines are important because they actually interact via their positive charges with the negative backbone in our sugar phosphate backbone that we have in DNA, and allows that we can actually get the different parts of the DNA molecule more closely to each other and can actually wind it around appropriate structures and so on.
but this winding and unwinding of DNA then also has other ramifications that are important for controlling cell growth and also cell functions. Okay, but this is not complicated. Okay, what is better understood is the role of another molecule that is based on amino acids. So this is not a biological amine anymore. Okay, so this is just another derivative of amino acids, and it's phosphocreatine. And phosphocreatine, as I said previously, briefly at least, is an important energy storage molecule that is of particular importance in muscle cells and in nerve cells, both heart muscle cells and also skeletal muscle cells. And it's based on 3 amino acids, as you can see here, namely glycine, arginine, and methionine. Okay. The structure of phosphocreatine is shown here, and we see which parts actually stem from what amino acid. And the synthesis starts in the kidneys, where glycine actually gets the so-called amidino group from arginine with the help of the appropriate transferase. So this amidino group here, the structure that is shown in this red box, is transferred to our alpha amino group in glycine to form something that we can refer to as a guanidinoacetate, because this is a classical guanidino group that you would also find in the amino acid arginine. Okay, but here it's linked to an acetate, so why not call it guanidinoacetate? Then this molecule in the liver is actually methylated, and the methyl group here stems from acetylmethionine, which we know is formed from ATP and methionine. So methionine also contributes 1 carbon to the synthesis then of this compound here, creatine. And creatine, especially in skeletal and heart muscle, but also in the brain, can be phosphorylated. And what is utilized here for the phosphorylation is an ATP. And ATP is converted to ADP, and we form this phosphocreatine here. Okay, and phosphocreatine is actually produced in the let's say cytosol / intermembrane part of mitochondria, and can then, when energy is required in these muscle cells or nerve cells, be converted back into ATP. So you could ask, why would we do that? Okay, why do we convert ATP or utilize ATP to produce creatine phosphate and then utilize creatine phosphate to produce ATP again? The reason is that by doing this, by utilizing ATP in the intermembrane space / cytosol, okay, to produce creatine phosphate, we produce an energy carrier that can be utilized when needed, but keep the ATP concentrations low in the cytosol / intermembrane space. Okay, so in aerobic metabolism, we produce ATP in the mitochondrial matrix. We learned about this, which can be transported out. So in order to still keep the ATP concentrations low, so that we continue breaking down nutrients, okay, we produce creatine phosphate, and then when energy is needed, we have a quick conversion of creatine phosphate into ATP, which can then be utilized for energy production. Now, creatine spontaneously cyclizes to produce creatinine, and is secreted via the kidneys. You may have heard about creatinine clearance before, okay, because this is widely used by doctors to assess your kidney function, because we produce relatively constant concentrations of creatinine, and so your creatinine concentrations in your urine should fall within a certain range, and if it deviates from the certain range, something's wrong with your kidneys. Okay, so that's one of the ways that kidney function and glomerular filtration primarily can be assessed. Okay, we are almost done. I think we have two more compounds to discuss that result from conversion of amino acids. Utilization of amino acids. One is glutathione. And glutathione, as you may know or may remember from what I said previously before, is the most important redox system in the cytosol. And it's actually a tripeptide that is comprised of 3 amino acids, namely glutamate, cysteine, and glycine. And this tripeptide requires the activity of 2 enzymes, 2 synthetases, and as the name implies, these synthetases consume ATP when they form covalent bonds between these 3 amino acids. What is, let's say, noteworthy is that the first 2 amino acids, glutamate and cysteine, actually are linked not via their, let's say, alpha functional groups, so the alpha carboxyl group and the alpha amine. Okay, this would be the thing that happens in the synthesis of other peptides that are synthesized by ribosomes. But what this so-called gamma-glutamyl cysteine synthetase, the first of these 2 enzymes involved in the synthesis of glutathione, does is it actually forms a covalent bond between the side chain carboxyl group of glutamate and the alpha amine of cysteine. Here, okay? And for that reason, we refer to this structure that is utilized here as a gamma-glutamyl residue. Okay. Whenever you have gamma-glutamyl, so it implies that you have a bond, amide bond, via the side chain carboxyl group, not the alpha carboxyl group. So we produce a dipeptide here, gamma-glutamyl cysteine, which is then utilized by the next enzyme, glutathione synthetase, again consumes ATP and adds glycine, forming a covalent bond between the alpha carboxyl group of cysteine and the alpha amino group of glycine. So the way I remember the structure of glutathione is, just remember it's gamma-glutamyl cysteine glycine. It's gamma, okay, so then you know what it's based on. So how can this molecule then serve in redox reactions? We have a sulfhydryl group here in the side chain of our cysteine residue. This can be oxidized with another such moiety, another glutathione molecule, to form the oxidized form of glutathione, where we have a disulfide bridge between these 2 cysteine residues. And then we can actually reduce this again to produce the reduced form of glutathione. We will actually see at least one example where this happens when we talk about nucleotide synthesis starting today. Okay, probably. So, last molecule that is based on amino acids that we're going to discuss is a tiny little molecule, okay, of great importance. This molecule here, known as nitric oxide. Okay, nitric oxide has a multitude of different functions. In a nutshell, you can look at it as an intra and extracellular messenger that has a very short half-life of just a couple of seconds. Okay, so this is not classical hormone. Also, it has lots of hormone-like functions, but it's not greatly transported in the bloodstream from some glands to secreting a hormone to the tissue where it's active. No, it's produced locally, okay, and actually has its function right in the proximity of where it was synthesized. And it has neurotransmitter functions, and among other things is involved in blood clotting, control of blood pressure, gastrointestinal motility, and many more things that are quite interesting. Okay. So how it's produced is shown here in this reaction scheme. There are a number of at least 3 or 4 different isozymes that are known under the name nitric oxide synthase. These are mixed function oxidases. We heard about mixed function oxidases last time when we talked about this mini electron transport chain that we find in desaturases. Okay, so we have actually two molecules that are being oxidized here. The arginine here is converted into a hydroxyarginine, so it's being oxidized, from which we cleave off the nitric oxide. And then also NADPH is actually oxidized to NADP+, and we utilize molecular oxygen. So very similar to what we saw for this mini electron transport chain last. Okay, so this concludes my discussion of amino acid metabolism. And what we are going to discuss now is the biosynthesis and degradation of nucleotides. So we're not just talking about the biosynthesis of nucleotides. So we are still talking about anabolism, but now we're also including discussion of the degradation of nucleotides. Why? Because we haven't done that yet. We haven't talked about the degradation of nucleotides yet, because nucleotides, as you may remember, and nucleic acids are not being utilized for energy production. So far, our discussion of catabolism was solely based on energy production. And this topic will then conclude our discussion of metabolism in the strictest sense, okay? But will lead us into what is typically referred to as molecular biology, okay? So I'll try to make you understand that it's just a matter of definition how you look at these things. But one step at a time, let's first talk about the biosynthesis and degradation of nucleotides. And remember, when we talked about the other biomolecules, I always gave you an overview, a review first, and I kind of do this here also, but it's incorporated in our discussion here of synthesis and degradation more or less. So if we look at the functions of nucleotides, quite naturally what comes to mind first is their function as building blocks of nucleic acids. Okay, and we'll learn about the various functions of nucleic acids then next couple of weeks. But we also saw that nucleotides are important energy carriers. We saw the role of ATP and GTP, and also activated intermediates such as sugar nucleotides or derivatives of lipids, also for activating these molecules for conjugation purposes. We saw that these molecules then also are important metabolic effectors, have important roles in regulating regulatory enzymes. And just remember this slide that I showed you at least once before that summarizes some of the regulation of regulatory enzymes that lead from glucose all the way down to the oxidative phosphorylation. And we have many steps here that are regulated by nucleotides AMP, ATP, AEP, okay, and either have activating or inhibiting activity. We talk quite a bit about the role of cyclic AMP as a second messenger. Just mentioned it a couple of minutes ago again. And we also saw a number of cofactor structures that included nucleotides, okay, in particular AMP, derivatives there. So, what is a nucleotide? I said this before, and you probably have heard about this in Dr. Baker's class. Nucleotide comprises three components: a nitrogenous base, a sugar, and a phosphate. Okay, and that's a simple definition that you may want to keep in mind. If we lack a phosphate, we're dealing with a nucleoside, S. Okay, so what you see in textbooks usually as a representation for nucleotides is something like that, where you have a sugar, usually it's ribose that is shown, linked to a nitrogenous base, and typically you see a purine or pyrimidine here. Okay, the structures are not shown here, just the names. And if we want to convert this nucleoside here, if we had a hydroxyl here, to nucleotide, you would link a phosphate. And typically what is shown is a phosphoester bond between the 5' carbon here and a phosphate, at least one, to make this a nucleoside monophosphate, where the phosphate is linked to the 5' carbon. Now, strictly speaking, this or a phosphate doesn't have to be linked to the 5' carbon. Okay, so if we had it somewhere else, we would be dealing with a nucleotide who are still having a nitrogenous base in the sugar. Also, not always the sugar is a ribose or deoxyribose. Here we saw in the structure of flavin adenine dinucleotide, or mononucleotide for that matter, that we can also have ribitol, okay, reduced form of ribose, and also the nitrogenous base. We'll see plenty of examples for that. Have seen examples for that already. Doesn't necessarily have to be a purine base or pyrimidine base. Just look at FAD again, the isoaloxacine ring, for example, or the nicotine amide ring in NAD+. Okay, these are not purine and pyrimidine bases, but still nitrogenous bases. But typically, most often you see a representation like this for nucleoside and nucleotide. And the nitrogenous bases that you usually see in textbooks and also in biochemistry classes as the classical representatives of nitrogenous bases are based on these two organic molecules, pyrimidine or purine. Okay, pyrimidine, just a heterocycle with a 6-membered heterocycle here, 2 nitrogens, the structure, and purine, which is composed of a 6-membered ring and a 5-membered ring here. And the nitrogenous bases that you usually see are derivatives of that, and I'll come back to that in a moment. But again, the typical nucleotides that you see in textbooks, then as sugar, have either a ribose, okay, or the reduced form of the ribose, deoxyribose, where we lack an oxygen here at the 2' carbon, excuse me. Now, if we look at this slide again real quick, because I want to mention something that you may not remember. The different atoms here that make the ring systems in pyrimidine and purine bases are numbered, okay, 1 to 6 or 1 to 9 here in pyrimidine and purine respectively. So in pyrimidine bases we go in a clockwise manner, starting with this nitrogen, 1, 2, 3, 4, 5, 6. In purine, we actually start with this nitrogen going anti-clockwise manner around this ring, and then in clockwise manner continue in that ring. So at this point, I usually blame some organic chemists some time ago for this numbering system. I don't know why it's done this way, but that's the way it is. But in order to distinguish the atoms in our sugars from the atoms that we find in the nitrogenous bases, we simply add these primes here. So the anomeric carbon here is the 1' carbon, so that we know that it's in the sugar. This is a 5' carbon. Okay, so when you hear about this prime, that's what it's there for, to distinguish it from the positions in our nitrogenous bases. Okay, so let's take a look at the most prominent purine and pyrimidine bases. We'll see that there are others also that are of great importance, but these are the ones that we most typically find in DNA and RNA respectively. So in deoxyribonucleic acids and ribonucleic acids, obviously in deoxyribonucleic acids we have this deoxyribose, and in ribonucleic acids we have ribose as a sugar component. So if we simply aminate, purine here at this carbon number 6, we produce adenine, okay? If we have amino group at carbon number 2 and oxo group at carbon number 6, we produce guanine. So these are the most prominent purine bases. We will see some others, okay? Then cytosine is simply a 2-oxo-4 aminopyrimidine. Thymine is a 2,4-dioxy 5-methylpyrimidine, and uracil is a 2,4-dioxypyrimidine. So if you look at these two structures of uracil and thymine, you see that there is only one methyl group different at the 5' position, not the 5' prime, the 5 position. And for that reason, you can refer to thymine also as a 5-methyl uracil. Okay. And this, as we will see, this methyl group is also incorporated in a particular way that we are going to discuss. I referred to this previously. When we produce thymidylate, actually we require tetrahydrofolate as a cofactor to add this methyl group here. Okay. So if you mess with dihydrofolate reductase and don't produce tetrahydrofolate, the active form of the cofactor, you mess with the synthesis, pretty much the methylation here of uracil to thymine at the nucleotide level actually, and don't produce thymidylate in sufficient quantities. And you mess with DNA synthesis. Okay, so a little bit about nomenclature, which is potentially confusing, but again, this is all stuff that you should know from other classes anyway. Just as a refresher, here we have the, let's say, the most prominent ribonucleotides and deoxyribonucleotides. So ribonucleotides at the bottom, deoxyribonucleotides at the top. We see names for the nucleotide, we see symbols, and we see the corresponding nucleosides if we lack the phosphate group. Okay? So, with regard to the naming, it's rather straightforward. May still be a bit confusing, but it's straightforward. So if we want to refer to the nucleoside that comprises ribose and adenine, we call it adenosine. Okay? I've used this term plenty of times in this class already. Now, similarly, if we have guanine as our nitrogenous-based leadership point, we have guanosine, and then we have uridine as a nucleoside, and cytidine as a nucleotide, or deoxythymidine. Okay, come back to that in a moment. Where it gets a bit more complex is when we want to express that we are dealing with a nucleotide. Okay, because there are two options for us, namely to just use the name of the nucleoside and then indicate that it's phosphorylated by saying monophosphate, diphosphate, triphosphate. So if we want to refer to this compound here, which is based on adenosine, which is shown in this red box here, we could call this adenosine monophosphate, and if we want to indicate where the phosphate is, we just say, for example, 5' monophosphate. Okay? We could also say adenosine diphosphate. It's a nucleotide, but we utilize the nucleoside name to indicate that it's phosphorylated. If we don't want to do this, okay, we could also simply call it adenylate. And adenylate is the appropriate name for the nucleotide. Similarly, guanylate for the guanine containing, uridylate, cytidylate, and strictly speaking, deoxythymidylate, but often enough we just say thymidylate. Where it gets more complicated is when you see symbols for whatever it's being used for. Okay, because often enough the symbols are being used for just for the nitrogenous bases, I suppose. Also, you don't often see that, but it could be used. So A maybe for adenine, but usually it's used for a nucleotide, okay? Like adenylate here, A, okay? Simple enough. And AMP would be used for adenosine monophosphate. And we all know that. We've seen that just a couple minutes ago. The problem is that A can also be used for the deoxyadenylate, okay? If you wanted to be more specific, you could say DA, with a lowercase d, front of the capital letter A, to indicate that you are specifically dealing with a deoxynucleotide, but often enough people don't do that. You have to deduce from the context what is really meant here. Okay, so don't get confused by that stuff. One thing that I would like to point out is that often enough you see something in textbooks like shown here also on this slide. It says, okay, we find thymine in DNA and we find uracil in RNA and no thymine. That's really an oversimplification, because we may find actually thymine or we do find thymine in certain RNAs where it's actually common, okay? And uracil you might find in DNA actually if you have a deamination of cytosine, but it's not supposed to be there. And then it's being recognized there. And it's recognized by DNA repair mechanisms, hopefully. But be aware of that. So this is an oversimplification, specifically with regard to something like that, saying that thymine is only found in DNA. That is not true. Okay, so yeah, let's at least start with the de novo synthesis of nucleotides, and I make it quick because what I really want you to know is just where the atoms in the purine ring system come from, which is something that was elucidated by John Buchanan in the 1940s and 1950s. And what he did was that he fed pigeons radioactively labeled precursor molecules and then determined where this radioactivity was found in uric acid that the pigeons would secrete. So pigeons secrete all the excess nitrogen in the form of uric acid, okay? Whether it stems from protein degradation or nucleotide degradation. And so he utilized birds to determine where these individual atoms in the purine ring system come from. And he found that this nitrate number 1 is actually stemming from aspartate. Carbons 2 and 8 actually come from formate, but this is actually introduced utilizing the appropriate tetrahydrofolate cofactor, and we see that these two nitrogens here, nitrogen 3 and 9, actually come from the side chain of glutamine. So we utilize appropriate aminotransferases to incorporate the nitrogen. And then these three atoms here, carbon 4, 5, nitrogen 7, all stem from glycine, and carbon number 6 here actually stems from bicarbonate or carbon dioxide. Okay, so that's something that you may want to remember. What I'm going to do, and we only have 7 or 8 minutes left, is I will talk about the reactions that actually produce purine nucleotides, and I don't expect you to know any details about this, at least up to a certain point, because this is a very tiresome process, okay? But I want you to understand how these nucleotides are being synthesized. And next time then we will talk about pyrimidine synthesis, and there are certain differences between purine synthesis, purine nucleotide synthesis, and pyrimidine synthesis. One, obviously, but one significant one being that our purine nitrogenous base never synthesized as the simple nitrogenous bases, but are actually synthesized already as nucleotides. Okay, so these purine ring systems are built up linked to a phosphorylated ribose. Okay, so it's not produce adenine first and then link it to something. Also, this is a different so-called salvage pathway, but we build up this ring system actually on a phosphorylated ribose, as we see here. And then, again, I'll go over this rather quickly because I don't want to bore you to death here. But we start out with a ribose 5-phosphate, which is produced in the so-called pentose phosphate pathway from glucose, and it's activated at the anomeric carbon by utilizing ATP, and we produce phosphoribosylpyrophosphate, pyrophosphate, or PRPP, very important molecule. You may have seen PRPP before. So we have a pyrophosphate here at the anomeric carbon to activate it, which allows us to transfer a glutamine from glutamine side chain nitrogen here in a aminotransferase reaction to this anomeric carbon. Okay, and this is our first atom that will end up in our purine ring system, and it's atom number 9, N9, this one here. Okay, so we'll see that we first build up this 5-membered ring and then continue synthesizing our 6-membered ring. A synthetase, again the name implies that it utilizes ATP, then adds these 3 atoms here, a little bit more, from glycine to produce something that we can refer to as a glycine amide or amide ribonucleotide. And all these names, which are rather complex, and I show you this occasionally, simply describe what structure you're dealing with. Okay, so if we look here at this molecule where it's relatively easy, we have a glycine amide. Okay, so we have an amide between our glycine carboxyl group and this nitrogen that we included previously here in our growing structure. Then we add a formyl group utilizing tetrahydrofolate and a transformylase here in this position, so we can call this a formyl glycine amide ribonucleotide. Simple enough. Okay, what do we do with that? We add another nitrogen coming from glutamine, requires ATP, requires a synthetase, and we actually add this here in this position, a formyl glycine amidine. This is an amidine structure. Then we utilize another synthetase, actually ATP, to catalyze a ring closure to produce a 5-membered ring, which is an imidazole, okay, which has in position 5 an amine. Why not call it 5-amino imidazole ribonucleotide? Then the next steps, which actually lead to this compound where we have added a carboxyl group, in mammals actually occur in one step. In bacteria, where this has historically been studied in more detail, actually requires 2 steps, namely first a carboxylation of this nitrogen here, and then a transfer of the carboxyl group to this position here. It, in bacteria, probably requires ATP. In mammals, probably not, okay? But end result is the same. We produce this compound, a 5-amino, 4-carboxy, amino, imidazole linked to ribose, linked to phosphate. Okay, ribonucleotide. Then our next nitrogen is brought in utilizing aspartate. So remember the different ways I discussed briefly, especially last time, that can be utilized to incorporate nitrogen into biomolecules. Here we saw already our side chain nitrogen from glutamine in 2 steps. Here aspartate is actually linked to this carboxyl group here. So you have a synthetase which requires ATP, and we produce a succinyl derivative of the structure that we had before. Similar to what we saw for the synthesis of argininosuccinate, in the urea cycle. Okay, and similar to what happens in the urea cycle, an appropriate lyase cleaves off fumarate, and we have incorporated our nitrogen here at this position. Okay, and we can give this the appropriate name, never mind. What then happens is that another formyl group with the help of tetrahydrofolate is incorporated, and then we have ring closure to close our 6-membered ring, and we have produced at this point a purine base, a nitrogenous base, which has an oxo group here in this position, and this base actually is called hypoxanthine. So you may never have heard of hypoxanthine. You've heard of adenine, guanine as purine bases. Okay, but there's another one, actually important one, at least one more, a couple more actually. Hypoxanthine, which is actually precursor of adenine and guanine. So the resulting nucleotide that is being synthesized here is called inosinate, or inosine monophosphate, or abbreviated IMP. And IMP is actually the precursor of both adenylate or AMP and guanylate. Okay, that's something that you should be familiar with here, how this happens. Because in order to go from our hypoxanthine ring system to the adenine ring system, we have to convert this oxo group into an amino group. Just look at this, okay? There are different ways that you could imagine how this could happen, how we could incorporate our nitrogen here, but yet again we utilize aspartate. Okay, aspartate actually is activated, or this position is activated, and then the amino group of aspartate attacks this carbon here. Interestingly enough, the energy to activate this group here, or this position here, actually comes from GTP, not ATP. Okay, I want you to remember that. But then we form this succinate derivative of adenine, from which we cleave off fumarate and produce AMP. Now, if we compare this with the synthesis of guanylate or GMP, we also use our hypoxanthine-based nucleotide, but we have to actually add an amino group here in this position. How do we do that? Similar to what we discussed last time for converting a hydroxyl group into an amine, where we first had to oxidize it and then had an aminotransferase reaction. Now here it's relatively similar chemistry, but first we have to oxidize this position. This is done with a dehydrogenase, which utilizes NAD+ as a cofactor to introduce an oxo group here, and we produce xanthylate, xanthine based, okay? And then we have an aminotransferase reaction, utilizing the side chain nitrogen of glutamine to convert this oxo group into an amine. And this, again, requires energy, but requires ATP. So remember that for the synthesis of AMP, we require actually GTP, and for the synthesis of GMP, we actually require ATP. Okay? So I would say that let's go over this real quick. I'm done in a moment. Okay. All I want you to see here is that for the regulation of purine synthesis, we have a number of regulatory steps that can be inhibited by intermediates or products in these reactions. So we have classical feedback inhibition. Okay, that's all I want you to understand. And I want you to realize that the de novo synthesis of purine nucleotides is extremely expensive. So depending on whether or not at this one step we require ATP or not, for IMP we require already 6 or 7 ATP equivalents, and then if we go to AMP 1 more, if we go to GMP 2 more. So remember that. And this will be important to understanding why then different type of pathway, so-called salvage pathway, is important to actually produce these nucleotides. Okay, I see you next week.